Okay, let's start. Uh, Good morning, ladies and gentlemen. My name is Saeed Nahavandi. I'm the chair for IEEE SMC Victorian chapter. And today it gives me great pleasure to introduce our special speaker, Professor Maia Angelova. And Maia is a professor of data analytics and machine learning and director of Data to Intelligence Research Center at the School of IT at Deakin University, Burwood campus. She was a professor of mathematical physics at Northumbria University at Newcastle upon Tyne in the UK from 2004 to 2016, and a lecturer in physics in Somerville College at Oxford Universities from 91 to 96. Maia's research expertise in data-driven modeling of complex systems, especially in healthcare, and a strong expertise in time series analysis, spectral analysis, and dynamical system applied to physical life and social systems. She also uses symmetry method and is an expert in structure and properties of material, carbon-based and magnetic materials, quantum optics, and quantum control. Maia's research is at the boundary between theory and application, and she is increasingly interested in translational research into the areas of health, medicine, and healthcare. With this short introduction, please join me to welcome Professor Maia to deliver her speech in the area of AI and time series modeling in sensing. Over to you, Maia.
Thank you, Sid. Thank you for the invitation and the kind words. And before I start, I would like to extend my gratitude and support to all businesses in Melbourne, because we are going to be open tonight back to norm or the new normal life. Okay. So can you see my screen?
Yes.
Okay, so today what I'm going to demonstrate is our research, mine and that of my colleagues, which uses a time series from different sensors to model behavior, properties, and other things of complex systems from different domains. So that's the title, and I will be basically trying to connect two things: artificial intelligence in the widest sense of its definition, which incorporates machine learning, data mining, network analysis, and all things that would allow us to understand better data from any size and any mode. The other thing is sensing. We have been increasingly used different data from localized sensors, such as sensors used to measure heart rate, respiration, brain waves, or continuous sensors, such as new materials used for smart floors, where the signal doesn't come from a single point, but comes from a continuous surface. And this is a little bit of background. This is the Data to Intelligence Research Center at Deakin University. It's a newly established research center which hosts 4 research labs. One is Data Analytics Lab, one is Mathematics Lab. We have Smart Networks Labs and Machine Intelligence Lab. So we have over 40 academics who do both teaching and research, who are actually experts in different aspects of machine learning and artificial intelligence in general. So this is what we do, and I would basically highlight the fact that we handcraft algorithms and models. We don't use black boxes. We sometimes do, but most often we need to really adapt what exists or develop new algorithm to handle the information hidden in data from different size and different sources. So we do develop data-driven models. So these I call bottom-up models. So these are data model-free models where we don't know anything about the behavior of the system that we're modeling, but we are learning the behavior from the data. And phenomenological or top-down models where we use system science, basically systems dynamics, and other techniques, including computational techniques, mathematical analysis, to try to derive a meaningful sort of information about the system. And these models are more useful when we need to learn about the causality. Like top-down model allow us to do this. Bottom-up model would give us the hidden patterns and would allow us to learn more about the system. And these are all applications like domain that we have worked so far, but we are open to new domains. I forgot to add here, I have also worked with a lot actually in UK with communication, optical communications, optical and wireless communication engineers. So these are my collaborators, and I've been around for a long time. Some of this research that I'll demonstrate today was spin-off by a European Framework Program 7 model from Marie Curie program, which was, I coordinated this research project, and this was one for a mobility project. So we had a lot of connections with countries outside Europe, including Australia, Griffith University, and South Africa, Mexico, Germany, France, UK, and Bulgaria. So the people who are in color blue and red, they are people with whom I linked during this project. And basically, the project was about models and technological solutions for improving and enhancing the quality of health. And that's where I actually moved from one field, which was more linked to material science and condensed matter physics, to another field, which was life systems. A lot more difficult, but a lot more exciting as well. So what I'm going to talk today is about modeling complex time series for objective evaluation, and I'll focus on things that I think I can show case studies. So this would be health and human performance. What we'll be using are hybrid models. So these are hybrid models between mathematical modeling, mostly based on dynamical systems and differential equations, but not necessarily, and machine learning models where we use machine learning to study the parameter space. And this is some of the models were for decision making, as we work a lot with DST group in Canberra. So we use artificial intelligence, and I would say artificial intelligence in the most general form. I also had, when I was in UK, I had a telecare robot of the giraffe series. So we use combination, artificial intelligence and mathematical modeling to model large and complex systems and time series, which are obtained from sensors. And this could be multiple sensors, and could be sensors of different types. And these are sensors that we have experience with from medical research, healthcare, and sport. However, if there are other data sources, there is nothing stopping us to extend this to other type of sensors. So the case studies that I'll show you today are classification of insomnia, acute and chronic insomnia from healthy sleep using actigraphy data. So these are data collected via accelerometer or actigraphy watch. Then glucose and insulin regulation for detecting diabetes 1 and 2. Then chronic condition support in elderly, telecare call centers. So this is the results from our European projects. And physical and cognitive performance in athletes. And I'll use an example from the Ironman competition.
Hi, Professor. This is
Yes.
Salakin here. Do you mind if I ask a question?
Yes. No, I don't
So,
mind.
so in that hybrid models, you mentioned that you use mathematical expressions and data-driven models, which is machine learning. So, are they in sequence? Do you use mathematical expression to massage your data and then feed that into machine learning model, or
I
is that
think the best way to answer this question is I'll show a case study, use case,
sure?
where this is illustrated. Depends. Sometimes we do it, depends how much you know about the system. If you know enough, you can use the mathematical expressions first. If you know nothing, you start with the machine learning, data mining to learn a little bit about the components of the system. But I have different examples and it will show you how it is embedded.
Sure, sure. Thank you.
Okay, so here, what type of sensors I'll be looking at? So basically, as I said, artificial intelligence I would use in a very widest sense, including network analysis, which includes complex networks, embedded networks and multiplexes, as well as machine learning, data mining, and some aspects of robotics. So in terms of what we use, and most of my examples would be with accelerometry data because this data are extremely noisy and very hard to model. So basically what works on this data can work on almost everything. So these are instruments that collect data for physical movements. So they used in sleep research a lot. Some of them are now used in medical research and also a lot in sports analytics. So what is shown on the picture here, this thing is like Philips free axial accelerometer. Free axial means that it has three dimensions, and one of them is the gravitational acceleration. And this one, the second one, is a Fitbit from the Fitbit series. Then we also have studied physiological signals collected with EEG. And these days we have portable EEG like gait data with sensors on the fit. So this is the portable EEG, and the first instrument is for ECG. So heart rate, brain waves, like I would say this is more like a physical movement rather than physiological, the gait. And very recently, we are just starting to work with a company which has produced sensing floors. So these floors are based on graphene, carbon type of material. So these are graphene layers. You know, graphene is two-dimensional structure, which is honeycomb composition of carbon atoms. So with this company, we're hoping to develop to develop it to a product where we can use it for elderly, like aged care facilities or rehabilitation facilities, not only to detect floors, but also to prevent floors, to detect falls, but also to prevent falls, because the material can be underlayered with soft materials. So this presents a real challenge because the signals don't come from a single point. There are multiple signals, but also it has potential, full potential of which we haven't investigated yet. So physiological I'll use physiological signals and for anything that comes from the human body, although it's not quite correct. But they have their challenge. First of all, they're very noisy. But the noise is something that we need to keep, because if you clean the noise of this physiological signal, then we clean the important information. So when we handle like signals from ECG, EEG, accelerometry, we need to incorporate the noise into the model. Another thing is that different sensors produce different type of time series. Like ECG, for example, is a short time series, while accelerometry is a long time series, and we cannot use the same analytical methods. For example, the trended fluctuation analysis would only work for a long-term time series like actigraphy, but not for ECG. So one has to be very careful what type of instruments from MATLAB, for example, one uses to analyze uh, the signals. So what we all know for this time series from school, from university, the easiest thing, or the first thing one would do with engineering background, is to go and do a Fourier analysis to try to find the Fourier spectrum, to try to find the periodicity. Some of these series would be periodic, but the periodicity could be hidden. And indeed, that's what I did as well when I started. And this is not a wrong move. So looking at the Fourier analysis would give us some initial information about the behavior of this time series. But it's not enough. Then there are many new methods developed: fast Fourier series, wavelet analysis, empirical mode decomposition. We also have contributed to development of these, I'll call them spectral analysis techniques, and more techniques are appearing every year. So here you can see three signals. One is the heart rate, one is the from breathing, and these signals are characterized with their amplitude and frequency, or the time point. One thing that is important, that we need to have more than 3 time points to analyze a time series. We have the minimum we have done is 4 time points, but it was really a struggle. So more time points we have, the better. And we convert this to frequency, and we can look at the signal into the phase space. So most of the models that we use don't look that much on the amplitude, but on the frequency of the signal. And we use the amplitude to clean the noise. So we put filters related with the amplitude of the signal, which is really how high the peak of the signal goes, and the frequency is linked with the time point on the horizontal axis. So here we have a physiological time series from the brain and the heart. And what is important to remember is that these signals are strongly nonlinear and oscillatory, because our heart beats continuously and periodically. If it stops, then we have a problem. And also the brain thinks continuously, and the waves are created periodically linked to the circadian rhythms. And when we sleep, we wake up and so on. So when we measure the power output from heart and brain, we can actually capture the power output trace, which contain a lot of very rich information. And as we can see here, this is basically from the brain waves, rapid eye movement. Like we see that we have, there is a strong link between them, that we cannot work in isolation. And that's why these days we talk about network physiology, where we consider different organs of the human body connected in a large network. So the measurements that we do measurements from brain, we do measurements from different parts of the human body, heart, wrist movements, leg movements, and so. This is how they look. You can see on the right-hand side of my screen different signals from different instrumentation. So this is what basically is a recording of one night in a sleep laboratory. And what we also use these measurements for is to look at normal or disturbed sleep. I'll show you one of our case studies to classify the type of insomnia. Also, we're looking at aging, change of function with aging, physical activity. We are looking whether the brain is normal or injured, or if there is a problem like epilepsy or another disruption of the brain flows. And all of this, basically, we can analyze with mathematical and computational methods. So the first case study is our work, which is basically my flagship project on acute and chronic insomnia. And so what we're trying to do, we're trying to develop pre-screening tools so people could actually identify the condition at home and go and see a sleep specialist like a clinician. The motivation for this, and we try to present it with this like animation or caricature on the left. People are taken to a sleep lab. In the sleep lab, there are lots of instruments and sensors attached to the person, and this person maybe can have a good night's sleep, maybe not. One thing that is known, however, for people with insomnia, when they change their condition, their bedroom, because they associate the bedroom with the insomnia, they have actually a very good night's sleep at the sleep lab. And this is another reason why polysomnography, which is the traditional sort of measurement for sleep research, doesn't work for insomnia, because people stay in the sleep lab for 1 or 2 nights. And sometimes they have a very good night's sleep, obeyed all this instrumentation around them, and the condition cannot be captured. That's why accelerometry or actigraphy, which measures wrist movements, has been used as a complementary tool in sleep research, and collection of data is usually for 1 or 2 weeks, where the condition of insomnia can be captured. In addition, like the measurements in the sleep lab are very invasive and expensive. One night in sleep lab in UK costed about £100. So it's about $200 per person per night. So you need $400, and you need funding to basically have at least 30 people with the condition and 30 people for normal control. So in addition to that, there are other motivations. So this is sleep is naturally occurrent transient behavioral state that is characterized with these things: reduced mobility, loss of consciousness, reduced responsiveness to stimuli. People can be affected by very old-fashioned and well-working techniques, such as listening to classical music, reading, or using of opium or alcohol, which I would not recommend for current dealing with sleep disturbances. But also sleep disturbances are linked with very serious health conditions, the one of which is depression. So if you have many sleepless nights, it's very likely that you will develop depression. And if this is not cured, that can go to much more serious things like chronic depression, etc. But as I said, polysomnography, which is the golden standard in sleep research, is not effective as objective method for investigation of insomnia. Currently, this is done by clinicians using the sleep diaries, where people record their behavior when they go to bed, what time they go to bed, what time the lights are out, what time they wake up. And as we know, this is very subjective. So whatever you do, you can't actually record correctly everything. So sleep diaries, they are very important tools in sleep research, but they're prone to mistakes. So what we're trying to do, we're trying to replace the sleep diaries with the use of actigraphy, to replace the use of polysomnography for detection of insomnia only with this sort of objective, non-invasive and very cheap type of measurement, namely measuring the changes in the body movements during the day and during the night for over 2 weeks. So the data, we work with a couple of sleep labs in Australia and outside of Australia. So the first time I started working with insomnia, and in particular acute insomnia, with my colleague, Professor Jason Ellis from Northumbria University, who is a sleep clinician. Then when I came to Melbourne, we developed collaboration with Monash University Brain Institute with the Sean Drummond Sleep Lab. We also developed collaboration with Sydney University, their Brain Institute, Sveta Pusnova and Chris Gordon Sleep Lab. And also we work with Charité University and Thomas Pencil Sleep Lab in Germany on sleep apnea. So also we have other collaboration. We work with Boston University and Plamen Ivanov on his network physiology lab. We work with Mexico, the National University and the Center for Complexity. We work with the Bulgarian Academy of Sciences and the Bioengineering and Biophysics Research Institute. We work with University of Exeter, Essex, the Sports Sciences in La Trobe, the Sports Sciences at University of Cape Town, Sports Sciences at Deakin, and the DST, there is a spelling mistake in Canberra, and DSI at Melbourne. So the methods for investigation, as I said, actigraphy is one of the most notoriously difficult signal because it's very noisy. So what you see here is the time series collected from two individuals. The blue signal is a healthy person. The red signal is the individual with acute insomnia. They're both same age. Young people, basically, today I'll show you results mostly for people up to age of 40. So what we have tried to do is we imposed on the signal Costinor basically analysis. So this is the broken curve. And you can see that there is some periodicity. And also you can see with naked eye, you can see that there is a significant difference between the people with acute insomnia, this region, for example, and normal sleepers. So this is the signal taken 24 hours a day. And on it, we developed a method called singular spectrum analysis, which basically detects when people are falling asleep, so they don't need to record the time. And as you see, it's not entirely the same as the Costinor. Costinor analysis is a traditional analysis used in chronobiology and anything that is to do with periodic behavior. But the Costinor analysis was not able to capture the small, subtle differences that we observed from the actigraphy signal between acute and normal sleepers. So the study protocol, we basically did the following. We analyzed the night behavior, we analyzed the daily behavior, and we analyzed also the 24-hour signal. So we analyzed the signal from all sides. For this particular study, because the sensors were not modern enough, today there is a new generation of Philips actigraphs that actually have a software embedded to detect when people fall asleep. At the time, these were data taken from 2009 to 2011. such instrumentation was not available, so people were told to go to bed at 10 o'clock, not necessarily to fall asleep, but to go to bed and leave bed at 8 a.m. What we did for the classification, we used binary model where we did not use any sleep diaries, so we did not know anything about the patients. So basically all nights of insomnia were labeled as bad nights, and all nights of healthy subjects were labeled as good nights. And in principle, this is a wrong starting point. But our machine learning model actually was able to improve this. So what we did analysis of individual subjects. And here I'll show you mostly the nighttime actigraphy. So what we had 24 subjects healthy and 21, so 24 healthy, 21 with acute insomnia. For them, we selected out of all 14 nights, we selected 7 nights where the data were available. So this makes for all subjects 323 nights for the healthy and 266 for insomnia patients. So basically, you call this big data. So the age was less than 40 because when you go over 40, aging imposes different challenges. So then because we didn't know anything, insomnia is actually very complex. Sleep scientists don't really know what is causing it. It could be multiple reasons. It could be cognitive reasons. It could be physiological. So we started from the data. So this is an example of a data-driven model. And also machine learning model. So what we did, we took the signal and we started extracting features. So we started with the standard statistical features like mean, standard deviation, root mean square feature. But then this was not enough. So we looked at the dynamical features. So we looked at Poincare maps, 2 features from them, their ratio, and also complex correlation measure based on the Poincare map. So these 4 are dynamical features. Then we looked at entropic features like sample entropy and sample entropy is one example, but there are also other types of entropy. And then the last 4 features are these typical features which sleep scientists will extract from sleep diary, and we extracted them from the signal. So sleep onset latency, wake time after sleep onset, sleep efficiency, and total sleep time. So this makes 12 features altogether, and then we decided to deal with noise. So we extracted them with no noise filters, and then we impose filters on the amplitude of the signal to deal with 20% noise, 40 and 80. And each time we extracted the same features. So we ended up with 12 times 4, 48 features altogether. When we started these features, you can see the last column, the AUC value, and you can see the p-value. So you can see that there is no single feature from all of those that we extracted, which has significance bigger than 65, 70%, which means that no single feature can describe the process. So what we need to do in this case is we need to put them together in a machine learning model and look for a signature. So we developed, and that's what I mean, handcrafted models. There are lots of classifiers available in different tools, but one has to be very careful because sometimes if you do automatic feature extraction, some of it is basically artifact. So feature extraction is very, very important actually. And my recommendation is to do it like step by step. So here we have our model. So this is a model which was published 2020 in IEEE Access. So the model is 2-step. The first level is the classifier, and the second level is multi-step optimization. So the first step, we basically use 2 types of from the shelf classifiers, such as random forest and vector support machines. And the results of this are not very different, but random forums performed better. And then we input these results of the machine learning model, we put them into the classifier, and we did multiple optimization to try to improve the results. So here are the results of our model. You can see that accuracy is good, but also when you do machine learning model, you cannot, very rarely you can achieve equal accuracy, sensitivity, and specificity. Normally, if you want high accuracy, you have to give up a little bit sensitivity or specificity. So without the optimization, this is what came up from the model. This is on the left side of my screen. So 68.59 was the highest accuracy we achieved. And the reason is because we started with a wrong hypothesis, and our model is very, like, it's a crude. We basically have a very simple hypothesis, which is also wrong. All insomnia people have bad nights, which is not true. Everybody has some bad nights and some good nights, and all healthy sleepers have good night always sleep, which is also not true. Everybody could have from time to time healthy sleep. So when we did the optimization, we actually improved the results of the model to 84.5. And here you can see the AUC curve. And I guess everybody knows what is AUC curve. It's an area under the curve, which is used to actually evaluate the effectiveness of the model. And you can see the false positive and true positive rates here. So what we can do, also we can balance the model. If we want more balanced model, so relatively equal sensitivity, specificity, and accuracy, we can use what the model provides. And we can do some choice of nights. So what we have observed for acute insomnia is that 3.5 nights is the optimal number of nights for data collection, and it gives us optimal type of sensitivity, accuracy, and sensitivity and specificity. So this is the second study that we did with different type of data for chronic insomnia. Difference between acute insomnia and chronic insomnia is in time where the condition appears. If you have bad sleep for less than 3 months and then it improves, the condition is considered as acute insomnia. However, if it goes beyond 3 months, it's classified as chronic. So in the chronic case, the data were also a little bit of challenge. We haven't had a project funded directly yet where we can collect our own data. So we use secondary data that have been collected for different purposes. So in this case, our colleagues from Monash University gave us data for 40 bed partners. So it's the chronic insomnia person and his or her bed partner, who also has a little bit of effect from the fact that the other person moves a lot. So you can see here again on the first graph here, the first like diagram, the red would be for chronic insomnia, blue for healthy, and you can see that there is a difference in the behavior. So again, what we did for this data, we repeated the same steps as with the other data. And here you can see the extraction of features, and the different colors show the different filters that we have put on the amplitude. So the blue one is without removal of the noise. Red is 20%. Yellow is 40 and purple is 80. So 20, 40, and 80%. And you can see that the statistical features are always prominent, and some of the sleep diary parameters which are extracted from the accelerometer, not from the sleep diary. And you can see that the dynamical features, they are not necessarily very prominent without the noise removal, but as we start removing the noise, the entropy becomes quite important. So the next picture here on the top right of my screen is our two-level machine learning model. So the first level is the first layer is the classifier. Again, we use the binomial 0, 1, 0 for chronic nights, chronic insomnia, night sleep, and 1 for their bed partners, which, as they say, is a wrong hypothesis to start with. And then the next layer is the optimization. So we use the optimization to improve the accuracy. At the bottom, the two pictures shows the performance of the two classifiers. One is the random forest in the blue, and in black is the singular vector machines. And we can see their performance, what I said, that you cannot have 100% accuracy and 100% sensitivity and 100% specificity. They vary. And here, the last, sorry, I'll go back to the previous slide. The last picture shows that the results depends on the time of, on the number of nights recording. We cannot have good results with less than 4 nights actigraphy recording. So this is the second model, and
Professor, this is Salakin here again. Can I ask one more question on that? The last example, this case study one, is this an example of hybrid model or is this a traditional
this
model?
one is a traditional machine learning model.
Sure.
The hybrid thing is only here when we use Poincare maps and different types of entropy. Sometimes we design our own dynamical features. So it's not, this is not embedded in the MATLAB. So all these features.
So, and that happens as part of feature processing, is it?
That happens as part of feature processing. So this is an example of bottom-up data-driven model where we know nothing about the system. It
That's
is
right.
actually semi. It's not fully unsupervised. It's semi-supervised because we make this assumption about the quality of nights, 0 for insomnia, but like bad nights, and good nights for healthy. So it's not. So it's a supervised machine learning model.
Sure. And your two-step machine learning model, because you're cascading two different models, did you look into how the error flows from the first model to the second model?
Yes. So the second layer is really optimization. So what we've done here is to validate the model. So we have taken all the subjects in a mix, and we take out one subject and we validate on the rest, and then we do another. So you bring back this one and take another one and another one and another one until you exhaust all 80 subjects for this study. So you do it, and then you basically validate the model on this.
Sure, thank you.
But you can build different optimizers here, and that's another way where you can put a layer of mathematical formulas because you can change the optimizer. Okay, so this is the second case study, which is an example of top-down phenomenological model. So this is a study of diabetes where our aim was to work towards artificial pancreas. So in this model, we look at glucose insulin regulation, which is a periodic process with delays in our models, which are dynamical systems delay differential equation models. They appear with 2 delays. So you can see here, these are true data recorded from a person, and you see the glucose time series and the insulin, and you can see that they are in, when glucose is full, like is its maximum, the insulin is completely depleted. So these are 2 periodic time series, that are out of phase, I would say. So the maximum of one corresponds to the minimum of the other. So when the glucose is depleted, insulin starts being produced. And the process is like those of you who know about control theory. You can see this is the control loops. And in our model, we use two control loops looked with the plasma insulin and glucose production. Some models can be simplified to look just at one of the loops, or other things that we're trying to do now is to add the brain compartment, so how the brain would regulate the plasma and glucose production. So because we know, because we have this graph, we have this diagram, we know what is happening to the system. So we can use the top-down phenomenological model, which is based on assumptions. What is the communication between the plasma, between the insulin produced in the liver and the glucose consumption. So the functions that you see, F1, F2, F3, F4, F5, these are 5 functions which would mimic the physiological behavior in the system. We don't know exactly what are these functions, but we are trying to give expressions for them, which are what we assume would be the right thing, and then we fit, and then we improve. So in this case, this is a model which starts with the mathematical expressions. So I is for insulin, G is for glucose, and the prime means first derivative or rate of change of insulin sort of consumption with time, and rate of change of glucose consumption with time. So here we have the glucose intake. So this is a model based not on what you are doing when you're given some sugar to drink and then people measure you for diabetes. This is in a normal behavior of a human body. So this is not even after food. This is a normal behavior after people have eaten. So here GFT is the glucose and tau 0 is the delay. All the other functions, Q, F1, F2 are physiological functions, which I haven't written here, just to make things a little bit more simple. So that's what we have when we solve this model, which is very nonlinear, and boundary conditions are nonlinear as well. So we cannot solve it analytically, so we solve it numerically. And we can see that we can actually mimic the production of the glucose with time. So the blue line is the simulation, pink is what is actually has been observed. So what we see here is that the glucose, and we look at the steady state of glucose and insulin. So G star is the steady state when the system has reached equilibrium. We see that the two things are actually behaving in the opposite way. So the glucose is the blue line, the insulin is the red one. And then we start studying the behavior of the function and how the system actually depends on the delay. We also want to know what is the insulin secretion and insulin resistance. And we would basically benefit from more experimental data here. So just to finish with this model, we are planning now to work. There is a group of physiologists at Deakin who are studying diabetes in young people who are not obese, but they have insulin resistance. And we hope to further develop these models as we have more experimental measurements. I believe I have to finish soon, and Sayed asked me to leave 10 minutes for questions, so maybe I'll stop here. I have 2 more case studies, but I would give our slides to you and I can answer questions. I wanted to touch on the Ironman, but again, I rather answer some questions.
Okay. Thank you very much. That was a superb lecture, and maybe we open the floor to our guests, and then either they can raise their hand or speak and then ask questions. Are there any questions from anyone? Yeah,
Hi, I'm
Hushiyar.
Shadi.
Yes, first Shadi and then Hushiyar. So we have two hands up. Yeah.
Hi, fantastic talk. It's great and inspiring. Thank you for that. Just a quick question now for the sleep studies when we attaching the EEG and the other sensors. So how do you deal with the amount of noise that comes from the movement of the body? Like if the person is not sleeping well, it will be very hard to extract meaningful EEG signals from after all these movements.
Well, what we do actually, we take benefit from the movement. So all these results that I showed you are taken not from the EEG, they are taken from the movements. So movements measured with accelerometer. So this is like the gravitational acceleration of the sleeping people. And that's how, what I said is a very tricky, noisy data. And our machine learning model is extracted from this data. We haven't touched the EEG, we haven't touched the ECG.
Yeah, okay. So how do you know that if the person is in what level of sleep? How do you know if the person is deep in sleep?
Yeah, for this classification, all we knew was the classification that comes from the sleep clinicians, whether people are classified as acute insomnia or normal. We did not need to go deep into the sleep architecture. Now, because we are working on mathematical model of insomnia, so we now are going deep into the sleep architecture. And so what we're trying to do, which I'm not too sure, this is the objective of my RCDP grant proposal. So I'm not too sure whether we will be successful to identify the sleep stages from normal people on acute insomnia as they come from EEG to match them with the actigraphy. So this is one of the objectives for this purpose of pre-screening, because in pre-screening, we are trying to use a simple model as possible based on wearable devices and to try to avoid EEG and ECG taken. But I don't know whether we'll be successful. We need to go because we work with neuronal populations for the models. So we need, for this purpose, we'll need to go and use the EEG. And at the moment, the model, it's not published. We are in early stages. We're looking at free neuronal population and we only look at sleep, non-REM and REM. We don't go into the details of the four states in the non-REM. But again, huge challenges because people with insomnia, their non-REM stages, they vary depending on what they have. If it's anxiety, for example, you get one. We look at the hypnograms, they are very different. If people have, say, diabetes and insomnia because of the diabetes, or insomnia as a first sign of diabetes, the picture is one. If they have anxiety or cognitive disorders, the picture is completely different. So basically, what we are doing now, we're trying to expand the two process model of Borbeli to include the third process, which is I call it like the behavior driver. So we look at circadian driver, at the homeostatic drive, and the third one would be everything that comes from the brain or everything that comes from chronic condition or illness, which we don't think we'll be ever able to identify, but we can mimic because we have very strong computational people, and we mimic basically what comes from EEG.
Thank you.
But we are not there yet.
So we have three questions, Hushiyar, and then Ahsan, and then Chalak, please, in that order.
Yeah, thank you very much, Professor Maia. It was a great presentation. Actually, I've got a question which might not be 100% relevant to the topic, but obviously still within the scope of AI. So I've got the human perception model, which is a transfer function, a linear transfer function, including some motion sensation threshold, which is obviously nonlinearities. And I'm going to just basically use that model to train the neural network to act like the same model. Currently, I've got using the time series data. So the inputs are all time series, different motion in terms of acceleration and velocities and position. So at the moment, we have got the problem of instability. Like I've got one other researcher in the team that he is aware of the problem that I'm discussing. So is there, we have used NAR and we have used a normal feed-forward kind of neural network. So obviously with the multiple layers. So have you faced a problem in terms of instability once you are trying to basically create a neural network model to act like that?
Your network, your neural network would not converge that type of instability?
The accuracy is high, it will converge, but when you test it, basically, for some of the signal, it's not stable, so it wouldn't act like the main transfer function. So I was wondering if there is any kind of, I don't know, time series neural network-based algorithm that you would suggest to use instead of what you're doing, or if you have faced this issue before, that you can suggest us something to basically do to tackle the issue.
Yeah, we face it almost with every model. So
Okay.
until I see the signal, I can't actually give you an answer. But it may be that you need to either perhaps either change the number of layers, or you may change the type of the network, neural network, not to be like back propagation. Maybe you need a convolutional.
Okay.
So I don't know what we can do. You can test different things. Depends whether your data, are they, do you have really big data, or they are sparse data, or do you have missing data? We at the moment face this problem with, we try to identify left foot from right foot with accelerometry. So we have a student working on it. We have been fighting with it for 6 months now. And we had to change the window of sampling. So this is another thing that you can do, like try different sampling window sizes. But we still, basically the work is now being finished and we are writing up. But it is a tricky process, and sometimes you may give us a call and share the data with us, or just show us your data and we can give you a little bit more advice.
Yeah, sounds great. So, well, at least I'm not the only one who's facing this issue, which is okay. So definitely, I've got your contact details.
Yes,
and we will be bothering you later.
Yes,
thank you very much.
Okay, a couple of quick questions, Aslan, and then Chalak, please.
Thanks, Maia, for your valuable presentation. And maybe the previous speaker touched a little bit. So my concern, not concern actually, I was wondering, like, if do you have any plan to incorporate the deep learning technologies into this kind of like sleep staging and this kind of pattern recognition things? Because that is, you discussed, that was purely classical machine learning approaches.
Yes, this one is classical machine learning. We do at the moment, with what I said, recognizing left foot from right foot, we use deep learning model. We use convolutional neural network, but we changed it a bit because it cannot, what is on the market in MATLAB cannot do what we really need. So with a master student of ours, we are actually massaging, I would say, the convolutional neural network And our problem is that our time series is very short. We only have a couple of trials for one person. So this is the challenge. If we had more data, then we probably don't need to do so much hard work to adapt existing tools. We're also using Siamese networks for the deep learning because they have been designed for small data sets.
Okay, okay. Thank you.
Thank you. Over to you, Chalak. Last question. Thank you.
Yeah. Hello, Professor Maia. Thanks for your wonderful presentation. I've got a question maybe related to your topic and can help me to solve one of my problem. I've got an application which is the combination of the image and time series data, such as face recognition and heart rate. I'm wondering, do you have any idea which hybridization algorithm can help me to solve my problem?
So you have heart rate and face recognition. That's interesting. We haven't done this yet, this combination, but obviously you need to, it depends whether you want to analyze the data simultaneously or you want to look at the face, the image processing separate from the signal processing.
It should be synchronously work with each other.
Synchronously work.
Yeah.
Well, my last example that I didn't have time to show was a on synchronization. This, unfortunately, I didn't have time to show.
Can you share it with me maybe later?
Yeah, we use, we didn't have, we had two types of time series synchronized. So this was ECG and respiration. And we used, so that's for the Ironman competition. We didn't have the images, but we use empirical mode decomposition. Classical, the method is from 1994 or 5. It was designed for earthquakes. So that's what we use to analyze the signal, but we haven't used any machine learning data. It's just honest engineering signal processing methods there, where we worked a little bit on the empirical mode decomposition. So it is with Hilbert transform. But we are going to do now, we need to combine facial recognition for some of the defense problems that we do with heart rate. Again, what I would suggest to call us, share with us your problem. You can show us on the screen and we can try to give you some suggestions.
Yeah, thank you very much for your answer.
You're welcome.
Thank you very much. Thank you, Professor Maia, for your superb presentation. We thoroughly enjoyed it. We really appreciate it as part of IEEE SMC Victorian chapter talk series to make yourself available and give such a very interesting talk. And there are more questions, but unfortunately we have run out of time, and I encourage individuals to contact you directly.
Okay, thank you. And I will send you my slides if you want to post them somewhere.
Sure,
and
we
we
will.
are open, everybody can call us and we'll try to help.
Great, thank you very much.
Okay.
That concludes our session until our next talk next time. Thank you, everyone, and thank you, Professor Maia, again. Thank you.
Thanks. Bye. Bye, everybody.
Bye.
Thanks.